So let's take a look at the brain structures that are helping us sleep and stay awake when needed. So we'll start off with waking and arousal. There are actually going to be two major pathways we're looking at. So major pathway 1, the first one we're going to be looking at, is abbreviated as the PPT/LDT. What you really need to know is that this pathway is in the pons, right? And it's associated with arousal. So let's take a look at where it is. So we can see here the large bump here on the anterior surface of the brain is the pons. And so we see that the PPT-LDT is right here in the pons. Now this area is inhibited when we're sleepy. And when it's active, it sends arousal information to the thalamus. And the thalamus then relays it to broad areas of the cortex. Because remember, the thalamus has all these sensory inputs that it then relays and sends out to the different areas of the cortex. And so it's able to sort of provide these inputs and get the cortex to become more active in case something is important and we need additional arousal. Major pathway number 2 is the locus coeruleus. Locus coeruleus. Coeruleus means blue, and that's because it actually is blue. It has a blue pigment. This structure, the locus coeruleus, makes most of the norepinephrine in our brain for us. So it's a major, by far the most major source of norepinephrine in our brain. And remember, norepinephrine is that neurotransmitter associated with arousal, right? So that makes sense. The raphe nucleus, not far, also in the pons, makes serotonin. And both of these areas then send their neurotransmitters, the norepinephrine and serotonin, out to the cortex. Both these pathways send outputs to the prefrontal cortex. The basal forebrain, which we see here, is going to be a major area for arousal as well, and the lateral hypothalamus. Now let's take a look at how activity in the locus coeruleus and the raphe nucleus correlate with different types of awake or sleep states. When we're awake, the locus coeruleus activity is higher. As we stay awake longer, more hours, we start getting tired, we can see that there is a corresponding decline in activity in the locus coeruleus. When we're in slow wave sleep, there's very little during REM sleep, and as we are waking up again, levels increase. In the raphe nucleus, its activity also seems to correlate more with being awake. Here's the awake state, and then as we are getting drowsy, you can see the levels decline. Slow wave sleep, levels decline. REM sleep. But post-REM, bam, this big burst here of activity in the raphe nucleus producing more serotonin. So again, both these areas are active when we're awake and alert, not when we're sleeping. Now let's take a look at the basal forebrain. So we see here that it is an area, kind of, if you think about where it is, it's in front of the hypothalamus, but in the cortex. So not deep in the brain, it's in the cortex. And this basal forebrain area also produces wakefulness and what's called desynchronization of the EEG. Desynchronization of the EEG allows for more information processing. This area also sends arousal to the prefrontal cortex. The hypothalamus is also involved in arousal and sleep. So a lot of structures, right, are involved here. So remember the pons, hypothalamus, especially the lateral hypothalamus, and the basal forebrain. Now, have you ever wondered why caffeine makes people alert and awake? It maintains alertness by blocking the receptors for a substance called adenosine. Adenosine is a metabolic byproduct that builds up in our cells. So remember ATP, you may have heard about that. So yeah, ATP, the A in ATP stands for adenosine. And so as our cells are active during the day, adenosine starts to build up. And that apparently is a sign that we are getting tired with the greater amount of activity. Caffeine then blocks those receptors in the basal forebrain area as well as in the hypothalamus. And by blocking those receptors, the brain loses one of the signals it would normally have to tell us that basically we're tired. So the difference between, say, an easy day spent having fun, lounging by the beach or something, and a hard day of studying, even though you're not physically super active if you're studying, your brain is active. And so you're going to have more of this adenosine build up, and you're going to be more tired and fatigued at the end of the day. Now, a dose of caffeine will make you feel more alert by blocking those receptors and losing that cue that you're tired. Now, you may have also taken antihistamines, most often used for allergies. So like Benadryl is a good example. Antihistamines can induce drowsiness by inhibiting the neurotransmitter histamine in the hypothalamus. And so histamine in the hypothalamus is normally involved in alertness. And so if we block histamine, we are blocking this important signal for alertness. And we're going to lose that signal. So that's going to allow us to get sleepy and drowsy. The pons are also an important source of what are called Pgo waves. Pgo stands for pons. The G stands for geniculate, as in lateral geniculate nucleus. That, remember, is in the thalamus and is involved in vision. The eyes send inputs to the LGN, the lateral geniculate nucleus. So, okay, here's our first cue that this might be involved in visual aspects. And from there, it goes to the occipital lobe. So the O here stands for occipital. So these are waves that start off in the pons, go to the thalamus, in particular the LGN of the thalamus, involved in vision, and then activate waves in the occipital lobe, again, the home of the visual cortex, right? These waves trigger EEG desynchrony, which is what we see during REM sleep. So these waves are associated with the beginning of REM sleep. So here we can see down in this recording, there's a recording of the lateral geniculate nucleus here at the top, eye movements here through the EOG, brain activity through the EEG, and then muscle tone and muscle activity in the form of an EMG. So kind of fun, right? We've done EOGs and EMGs, and then this lab, for this chapter, we will do an EEG. So let's take a look. We start seeing these PGO waves. Bam, 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 here in the lateral geniculate nucleus, right? Notice that as we transition to REM sleep, we start seeing these rapid eye movements. Start seeing activity in the electrooculogram, the EOG. Meanwhile, the EEG has become desynchronized, and so now we're not seeing these big synchronized spikes of activity where all the neurons are firing together and going positive, negative, positive, negative. Instead, each of the neurons is doing its own thing, not allowing for a big buildup of positive or negative activity, but just sort of balancing it out and keeping it down towards the middle, so low amplitude. Meanwhile, the EMG, which at first had some muscle tone, hey, goes totally flat, no muscle tone. And so we can see that here we are entering REM sleep, where there's activation of the eyes, and we see movement of the eyes, but complete loss of muscle tone, enabling us from being able to move. So it stops us from being able to act out our dreams during REM sleep. Kind of a good thing, really. And we see that there is greater brain activity through the desynchronization of the EEG as well. And we see that the Pgo waves have become more common. Now, why would Pgo waves involve the lateral geniculate nucleus and the occipital lobe? Both those areas are involved in vision, and yeah, dreams are pretty visual, aren't they? So that's probably what's going on there. At least that's what people think. There is something called Rem Behavior Disorder. Rem Behavior Disorder is what happens in some cases of brain damage. And the brain damage may involve the magnocellular nucleus of the medulla. This area of the brain normally is what's required to produce this REM atonia, the complete lack of muscle tone. So this is important because, again, that's what stops us from acting out our dreams, right? In REM behavior disorder, with that type of brain damage to that part of the medulla, we lose that, and we're no longer essentially paralyzed during sleep. And so we do have examples of individuals who, when they enter REM sleep and start dreaming, they can act out their dream and they can thrash around really violently. Normally when people are in REM sleep, there's actually no movement of their body. The only parts that are moving is the eye movements. But the rest of the body is actually being actively paralyzed. Now, you may have heard, and I hope it hasn't happened to you, I've heard it's scary, but you may have heard of people waking up and not being able to move. That can happen. And it's because the atonia, REM atonia, has not let go, but the person is already waking up. And so it might lag a little bit behind, and the person might become conscious and wake up and be unable to move their body for a while. And again, I've heard it is kind of scary. Now, once again, the pons is important here in sleep. The sublateral dorsal nucleus of the pons governs the switching in and out of REM sleep. So seems to be involved in triggering these Pgo waves, which bring us into REM sleep and then getting us back out of REM sleep so we can cycle through our different sleep stages.